Thanks, Jonas, for the introduction, and thanks everyone for staying to the last talk. And I'm going to take a few minutes to cover a couple different topics, and I'm going to start with an introduction to the two roles that I have that you see listed at the top. CSO for Genomics at Discovery Life Sciences, and then currently still a faculty member at HudsonAlpha Institute for Biotechnology. And so what began as the Genomic Services Lab at HudsonAlpha has now recently become HudsonAlpha Discovery. And so essentially I arrived at HudsonAlpha about 10 years ago as a faculty investigator. As a member of their faculty, started a services laboratory, and then as that's expanded over the years, we realized that there was a number of challenges in kind of who and how we worked with, particularly as we tried to bridge biopharma, academic, NIH funding, et cetera. And under the nonprofit environment, it kind of became clear over the last few years that there were a lot of opportunities if we could just kind of figure out how to best bridge those gaps. And HudsonAlpha as an institute has always had a strong economic development effort. So there's 42 biotechnology companies on our campus. And what we essentially did over the course of the last year was partnering with one of them, Discovery Life Sciences, to create the very creatively named HudsonAlpha Discovery division, which is essentially a combination of the best parts of the nonprofit side with a lot of the business support aspects of the formal company, Discovery Life Sciences. And so now we're HudsonAlpha Discovery, and I got a fancy new title and also got a couple new bosses, which is always great. And but the great parts are we are now able to take advantage as an institute the size of HudsonAlpha that's not affiliated with a hospital or a medical center formally. We are always left with the question of how can we best acquire samples, how can we best characterize those samples, how can we appropriately power high-scale genomic studies, and the relationships with Discovery now really robustly enable that. So the Discovery Life Sciences has a global clinical site network that has nearly 300 clinical sites across dozens of countries around the globe, and so we can very rapidly acquire samples with deep phenotypic information on each one through this clinical network. And so what this collectively gives us, and I'll just kind of skim through this in the interest of time, is now an ability to have a large biorepository, a global clinical network, full characterization services including immunohistochemistry, flow cytometry, and then of course what my group enjoys doing the most, sequencing and bioinformatics services. So we kind of aggregated all these together under that HudsonAlpha Discovery capability. I do want to make sure that I don't give the inappropriate impression that HudsonAlpha as an institute was acquired by Discovery. It's not it at all. In fact, it was really the formation of a new entity combined with one of the biotechnology companies on campus. So the institute itself still remains the autonomous nonprofit research institute. All of my faculty colleagues continue to run their research operations in the same way they did before. And I'll, in fact, show some of the work of some of those individuals during this presentation now, and be sure to acknowledge them appropriately. So, and summarizing HudsonAlpha the Discovery under through a marketing lens looks like this: is that we really now have an end-to-end capability of going from build through isolate through analyze to understand, and I'll touch on a few of those things as we go through the presentation. So as a brief outline, talk about some of the results from the Sequel II Early Access Program that was run at HudsonAlpha. Also discuss some of our long-read sequencing and oncology applications, then finally where we're striving to go with some long-read sequencing and population genomics at the very end. So my lab's experience with long-read sequencing has existed for a number of years, and in fact still continues to actively use the RS2 instrument. So HudsonAlpha currently now has 4 Sequel IIs, one RS2, which is still running in support of Wave Life Sciences' Huntington's project, and that is best summarized as Wave has an amazing technology and capability that they are currently analyzing under clinical trial now for the treatment of Huntington's disease. And one of the requirements of that clinical trial is the ability to phase 2 SNPs on either side of the expanded repeat. And so in the RNA transcript, they're about 11 kb apart in total sequence. And so we needed a mechanism that could robustly phase those SNPs, characterize the length of the repeat, all essentially in one single read. And obviously a long read approach is the most appropriate way to do that. And when this started, it was when the RS2 was available, so it was pre-Sequel 2. And given the regulatory and experimental design and other considerations, it's remained on the Sequel II since, and has been actually been a great success Fortunately, about 75% of Huntington's patients have one or both SNPs in phase with their expanded allele, so it is a very high percentage of patients do qualify. But because of the way the program works, it is an absolute requirement to have that detailed and accurate genetic characterization run correctly. And the only other piece of data I'll mention from this so far from a technology perspective is the identification of the SNPs, the length of the repeat is done with there's essentially a highly redundant set of assays that are done with that. Sanger sequencing, 2 different PCR assays to look at the repeat length, and then finally, of course, the long read sequencing to actually do the phasing. So we kind of already know the right answer, but it did give us an opportunity to ask, if we set aside these other technologies, how accurately would just the long read assay detect the SNPs, phase them, and detect the length of the repeat? And it's essentially 100%. There's been a couple of samples that fell out for various technical reasons, but most of those are actually on the sample preparation side, not the sequencing side. So it's been, again, quite a successful effort. More recently, the HudsonAlpha Genome Sequencing Center, which is run by a couple of my colleagues at HudsonAlpha, Jane Grimwood and Jeremy Schmutz, had an opportunity to oversee the early access program for the Sequel II, and since that program, they've brought on 2 more sequels for a total of 3, and then my laboratory currently has a single Sequel II, so we have, again, a total of 4. Jane and Jeremy's lab is extremely experienced in sequencing and assembly of de novo plant and mammalian genomes, and in fact, much of their focus now continues to be on agricultural genomics and de novo genome sequencing and assembly in both in unsequenced samples as well as in sequencing of cultivars and very, very challenging genomes. So things with extremely complex structures, multiploid genomes, et cetera. So they're truly world experts in this. And they're, to be clear, their operations are within the HudsonAlpha nonprofit research, don't have anything to do with the HudsonAlpha Discovery efforts that I mentioned earlier, just to make sure there's no confusion. So the next several slides, you'll see at the bottom left-hand side of the slide that HGSC logo to clearly indicate work that came out of this early access program. So shown here is a timeline in terms of yield per smart cell in gigabase, which is on the y-axis, and then on the x-axis, the annual time from 2017 through 2019. And you can see a couple of indications of when the early access program started, when commercial release started, all the way through current. And so you can see that even with the significant increase in output per flow cell with the early access program, as you see highlighted there, and then another bump at commercial release, it's been a pretty, still a continual rise in output per flow cell since the commercial release, and this is certainly continuing to develop. So if we look at this from now focusing on the CCS HiFi sequencing, both in terms of raw yield shown on the left and then CCS yield per gigabase shown on the right, and again, just highlighting the 30 gigabase mark, which is shown there in red, and then when the Sequel II collaborative program initiated, which is shown on the little bar at the top quite recently, you can see, again, some of the same timeline indications. When Sequel was released, when the Sequel II early access program started, or essentially the 8M flow cells, and then that a little more granular indication of that continual rise on a per flow cell basis that we see over time. A couple of quick notes is that increasing the insert size from 12 kb to 15 kb with the new chemistry definitely led to an increase in the CCS yield per smart cell to about 30 gigabases. And then another technical note is that tight sizing of library fragment on the SAGE ELF is actually a fairly important technical note for driving this performance in the right direction. And then if we look at the number of passes versus the number of reads and kind of look to see where that peak is, certainly if you increase the number of passes, you can improve the error rates on the CCS HiFi reads, but there's a peak of about 5 to 6 passes that we're seeing here, as you can see, in three different experiments that's shown as part of the early access program when you look at that overall performance. And then if we look at the error rate, in this case it's an indel event per kb, and then the number of passes, you can see you quickly plateau. You're coming down to the indel error rates per kb is starting to drop off significantly by that 6 to 7 pass peak, and then certainly by 10 or 11 passes you're really plateauing and not gaining much additional error correction information by doing continual passes, to give you an idea of that balance of how many passes around since we're in Alabama, how many passes around the NASCAR track do you do before you start having a diminishing return in terms of the error rate? And then finally, summarizing, and this is a region of the human genome, so this isn't a plant genome. If you look at CCS calls just through standard analysis pipeline via the same call, same alignment characteristics on a pileup in this IGV view with Illumina, not surprisingly, for some complex regions of the genome with different structural repeats, et cetera, you can clearly see a resolution and a overall read per read performance and kind of extrapolate both of those from this figure to see that improved performance in complex regions. So we kind of wanted to ask, what other applications can we begin seeing glimpses of performance that would be analogous to and complementary to what we're using short read applications for? And certainly one of those areas of interest is RNA sequencing in IsoSeq. So with short read sequencing, it does an amazingly efficient job of counting transcripts and getting to a dynamic range and a level of resolution that's very empowering. But if you're trying to also characterize RNA structural analysis, fusion genes, and the like, there can continue to be algorithmic challenges, and it's quite analogous to some of the opportunities with structural variation in the human genome. And as you can see in the left-hand panel, we look at raw yield per gigabase from sequel to sequel 2, you see a significant jump in yield, and that, of course, is accompanied by a significant jump in the number of reads. And if you see, again, 4 million reads is highlighted, which is now becoming reasonably routine on the Sequel II instrument, which is now approaching a level of resolution per flow cell in terms of read count that it makes RNA sequencing reasonably attractive from a discovery perspective. So we wanted to ask a fairly interesting question. And so the next couple of slides, not this slide, but the next slide after this, I'm showing you data that's literally hours old. And so it is definitely not fully analyzed, but I mainly wanted to be able to show it for the potential. So while one of the things my laboratory has focused on both pediatric and adult cancer for a long time, and we've through discovery we have access to about 10 million fixed formalin paraffin embedded blocks. And we wanted to ask, can we get better quality nucleic acid out of those samples? And so without going into the great detail in this presentation of how that's done, I wanted to show a comparative analysis you see here. So on the left-hand panel is a collection of 79 samples that were done with a traditional RNA isolation. Actually, it's a combined DNA RNA isolation. So we are isolating both nucleic acids out of these samples, typically 2 or 3 10 micron, 5 to 10 micron sections. And what you're looking at is the percentage of sample, or the DV200 value for each of the samples, or the percentage of RNA molecules that are 200 nucleotides long or longer. And then the bar that you see, the red bar, is indicating 20%. So we've used 20% as a cutoff metric for quite a while, because it's right around that 20% metric is where you'll get a reasonably accurate, relatively low noise threshold for a traditional RNA-seq experiment. And anything below 20%, still workable. I mean, you can still get data from it, but generally speaking, the technical variation and the overall bias in the experiments can be a little bit high. And so we have looked at roughly 30,000 samples total, which are now split about half and half of them pre this protocol, half of them post this protocol. So we have a very good perspective on, are we increasing biological resolution? And not surprisingly, the answer is a strong yes. But I was kind of giving some thought to, with the output on the Sequel instrument, can we apply IsoSeq, to look at the detection of fusion genes and just kind of ask, can you do IsoSeq on FFPE material? So a fairly simple question, but obviously a little bit of a complex background. But if you look at the 90 out of 90 samples that had DV200 samples that are greater than 20%, you actually see that the mean is probably 55 or 60%. So they're reasonably long molecules. They're certainly not intact. That's the probably an important take-home message. They're certainly not intact RNA, but they're much, much longer than what's enabled without this. So we kind of first looked at the IsoSeq process in general, and this is just summarizing the entire informatics pipeline, but it's mainly a reminder to state that the traditional IsoSeq protocol starts with a poly-A or an oligo-DT prime cDNA synthesis, and then the expectation that you're getting full-length transcripts to do the final consensus and isoform level clustering. So we know that this particular process is not optimized for the type of RNA analysis we're doing, which is why the next slide that shows some of the analysis is very, very cursory. That combined with the fact that it's a relatively small or very, very short period of time since we've had some of the data. So I actually had a conversation with Andy Larea, I think 10 days ago or so, saying, hey, I want to try this. And at the time, the 3 sequels that we had running were booked. The 1 sequel that's in my lab is not quite yet validated. So I said, if I get the libraries prepared in a week, can you guys sequence the libraries? Unfortunately, PacBio said yes, and so what you're looking at now is the results where we took 11 RNA samples from FFPE tumor samples. These are the amplified cDNA plots, and I'll just draw your attention to the long tail that you see on the kind of going from left to right, which is indicating the higher molecular weight RNA that's present. It's definitely not in the majority, but it is certainly present. We used about 100 nanograms of FFPE RNA to generate cDNA. The standard IsoSeq methodology, we tried that first just to kind of see how much of it had poly A tails, how much was intact. It did not work. So we ended up coming up with a modified protocol that supports a range of RNA qualities. It's still under optimizations. It's definitely not quite ready for prime time, but we're pretty encouraged by these early results. And then so between that long tail as shown here, if we look at the higher statistics or a little bit more details on these samples, a couple of key things. If you look at the DV200 scores, you see these are all in the high 88% range, but notice the RIN scores, which is just to the left, are all still fairly low, except for one sample that had a RIN score at 8.5, which is probably artifactual given its relative performance that we saw after the fact. But then if you look at the cDNA yield was good. And then if you look at the different bins, cDNA concentration in 1 to 11 kb, percentage that's been 1 to 11 kb, they're reasonably variable, and that was a good thing as far as this test. And then finally was the PacBio IsoSeq library concentration, and then the final library yield, and then the library that we saw that was at least 500 base pairs out to 11 kb. So you still see clear indication that these are not intact RNA samples. But we also thought, given the long tail that I mentioned earlier, given some of these statistics, well, we'll just do a really careful size selection on the samples, and then we'll bias the reads to those longer fragments. And kind of shown here where you can, again, see those long tails in the libraries. So the IsoSeq libraries were prepared using the SMRT Express template kit 2.0, some slight modifications, nothing major. And then sequencing was performed at PacBio on the Sequel 2. So our first impression was we'll just size select the libraries. And that actually just, in summary, turned out to be a terrible idea. That it looks like putting in everything that's in the library actually gives a better yield on the sequencer. So either that or it's more indication. These are early days in this method, but with the following, again, very cursory analysis, I think there's at least reasonable things to be quite encouraged. So we have probably about 3 orders of magnitude lower reads than we'd want for the sample at about 40,000 on this particular flow cell. But it was one that we didn't want to cherry pick the best, didn't want to cherry pick the worst, so we kind of picked one in the middle. And but importantly, of those modest number of reads, which is shown with the asterisk, we did see a little over 90% mapped to HD38, with almost 90% of them mapping within genes, and then about 13% mapped to intergenic regions. And then the average read length was mapped read length was 302. Longest read was 8.5 kb, and we had 209 reads greater than a kb, and a little over 3,000 greater than 500 base pairs. So again, still indicating that there's clearly some area for optimization, but we're also able to do a reasonable pass at long read sequencing out of these FFPE samples. And if we ask what is the number of reads above 300 base pairs? So kind of, doing that contrast of, number of nucleotides detected with a paired 150 base pair run, or even at a paired 100 run on a short read platform, we're seeing about a little more than half of the samples are coming in at read lengths at greater than 300 base pairs. And in fact, the average after trimming is 435. And then fortunately, it does look like this is quality data. We're not looking at noise, and we're not looking at things that are artifacts of the cDNA amplification process or any of the library preparation process, because a full 93% of the mapped protein coding genes, and then about a little under 8% mapped to nonsense mediated decay areas. So an overall encouraging data set with the total number of transcripts whose expression was detected is a little over 2,000 in these samples. So we're obviously continuing with this analysis across a larger number of samples, and we're looking forward to doing this more, but I think it was a reasonably exciting opportunity to get longer, higher molecular weight RNA out of FFPE samples and start looking at some of the biological applications for this, and of course taking a more rigorous approach to this analysis, and looking forward to tweaking the pipeline a little bit. So maybe continuing with what else can you use long-read sequencing for that was traditionally in the short-read sequencing space. So this is an example from some colleagues and collaborators at St. Jude that we work closely with as they're part of our, together we have a Gabriella Miller Kids First Sequencing Center, and we use long-read approaches as part of that center. And so some of St. Jude's data is shown here. If you look at the PacBio on the B column below, PacBio on the top, Illumina on the bottom, and what makes this significant is the HBG1 locus. HBG1 and HBG2 have about a 90% sequence identity between them, but all of the important polymorphisms for sickle cell disease are located in HBG1. And so if we look at the targeted analysis of that region, the arrow at the top shows where a key polymorphism is called out of the PacBio data very accurately, but is an area, because of homology to the HBG2 locus, is an area of struggle with just the short-read data alone. The targeting approach is indicated in the cartoon on the top. It's non-overlapping biotinylated oligonucleotides. There's a shearing that was done around 6 kb or so. And then not surprising, the read length came in at about a mean of 4.5, exactly right where it was expected to be. So overall, from a technical perspective, this was a very successful experiment using a hybridization-based targeting, and then other approaches certainly exist using a CRISPR-Cas9 or other approaches to target regions of the genome that you saw a little bit of in the last talk. But again, leveraging long-read technologies for not only RNA sequencing, but now targeted sequencing. And then, of course, the general use of long-read in the analysis of genomes that have highly complex aspects. So again, an example from St. Jude, a little bit closer to their pediatric cancer genome program, where you're looking at 2 different samples with Ewing sarcoma, which is known due to the DNA repair defects to have substantial structural variation and complex rearrangements that are shown here. So again, important analysis or important capabilities for long read. I'd say in some very elegant, very, very nice publications, one shown here from Jesse Dixon out of Feng Yu's lab, and indicating, and without going through this figure in detail, it's really just to use a few different figures from that paper to indicate the application of multiple technologies all distilled down to show one thing, and that's cancer genomes from a structural perspective can be, and in fact are often, very complex. And you see that on the left-hand side in the D panel with the different structures that are shown in the cartoon there, and then on the opposite side with some different technologies that show, again, similar structural variants and a lot of complex rearrangements in these samples. So again, warrants the idea that this integrative analysis and integrative detection in cancer genomes is clearly an area that we have to pay attention to if we're trying to truly decipher the full spectrum of mutations and genomic effects and how we have the transformative events that take a cell from not only from normal to pre-malignant and then on to metastasis is of critical importance. And so that's one of the reasons we've presented and are now currently applying long-read technologies in the Gabriella Miller program for the current batch of cohort samples. In addition to cancer samples, we can also ask, what is the overall population structure of structural variants? We have now the ability to very robustly and accurately analyze not only not thousands of genomes anymore, but now the technological capabilities to analyze truly millions of genomes and truly understand the structural the population level single nucleotide variants. And Mark Chazen and a number of colleagues put out a fantastic paper in Nature Communications this year. The reference is at the bottom. And I think one of their very elegant figures is shown in panel B, or it's one of my favorite figures from the paper. And what you're looking at is kind of going around clockwise from 12 noon is insertions of different sizes, and then as you continue around from 6 o'clock back to 12 noon again is deletions of different sizes. And the different colors represent what's observed with different technologies. And so again, you're looking at an integrative analysis of this, which I think is critically important in that a single technology with the current totality of our ability to work with genomic data is not yet giving a clear and consistent picture. But it is showing, I think papers like this, that is, again, a tour de force as far as work, is showing a lot of important things. And what you see here is in green is the PacBio unique structural variants that are shown. So you see there's a high percentage of green, but then there's also The pink at the top is Illumina unique. And then what you see towards the center of the circle or the different colors is when you now have an agreement or overlap between the different technologies. So it's showing that across a wide range of sizes and across a wide range of genomic positions and structures and sequence contexts, you clearly get value, and there's added value of doing the integrated analysis. And so we're fortunate to now be able to have a role in the All of Us program, which in summary is the goal of sequencing 1 million genomes over the next 5 years or so, with 3 national genome centers at Baylor, Broad, and University of Washington in Seattle, and with a goal of doing the majority of that work on short-read sequencing. And then most recently, HudsonAlpha was awarded a 1-year award to perform some complementary long-read analysis on with a goal of achieving about 7,000 whole genomes of sequence over the next 12 to 18 months or so, which we're particularly excited about. We're thrilled to be part of the program, and we're very, very encouraged by the technologies that are available. And what you see here is a little bit of modeling at where we think things will land, and this is assuming about an 80% per base accuracy rate, where you look at structural variant recall shown on the on one axis, and then the individual as a population sequenced on the x-axis, and with about 7,000 samples or so sequenced, we're expecting to have an ability to detect down to probably about a 0.1% population allele frequency for structural variants. And so this will, in summary, will allow us to ask a simple question that will no doubt have a complex analysis associated with it. It is, what types of variants are best detected in population scale sequencing with short read analysis, and then what types of variants are potentially best detected with long read analysis, and then probably most importantly is what lessons can be learned from from those overlaps and from those complementary efforts to best understand the goals of all this, which is truly to understand human variation and its effect in the range of normal, how it applies to disease, and then to truly march us towards the ability to step beyond single gene, single effect types of analyses. So with that, I'll acknowledge my team, which is shown on the left-hand side, as well as really critical collaborators and colleagues, both Andy and Primo at PacBio for very rapidly supporting both the sequencing and the analysis of the data. Some of the data you saw, our colleagues at St. Jude, which are key partners in the sequencing center that we're thrilled to be a part of. And then I don't have quite pictures of everybody on my team, mainly because you see many of them look misshapen as I tried to resize the pictures. So rather than inappropriately size anybody else, that's a subset of the team. But they're all listed there, and we're thrilled to be moving forward in these new directions and kind of new structures with HudsonAlpha Discovery. So thanks for the time. Really appreciate PacBio giving me the opportunity to share some of the results of our early access program, as well as kind of where we're trying to head in both population genomics and some of the RNA sequencing. And I'd be more than happy to take any questions.